Dear organizers, dear participants, good afternoon. The topic of my presentation is called Working with Safety Signals. I will speak about main principles of working with signals, including automation of this work, and also whether this work has to do with real world data, real world evidence. There are some publications about signals, there are some standards, and there is real practice of working with signals. For those who do not know this theme very well, I would like to say that most statements of my presentation have been published in articles that I have written. And all these articles on signals were written together with Dr. Yanovskaya and other doctors who had been dealing in signals for a long time. These articles show how doctors should work with the signals to minimize risks for patients and for the industry. And some slides of my presentation show or give a reference to the articles that we have written. You know, the term signal has a definition. Those who work in pharmacovigilance know that this term has changed. The qualitative criteria has been removed. The renewed aspects have been renewed. So anyway, signal is an information from one or several sources about a higher level of reliability of causal connection between the drug and its use, about which nothing had been known before. That's why the developing companies should know signals and should work with them very actively. If there is no work with signals, or if this work is incorrect, so the inspection of your pharmaca survey will find all the discrepancies. You know, for a long time we worked with signals manually. Many institutions continue working this way. But now the work with signals is becoming more and more computerized. And it is necessary to introduce automation into the work with signals, and no matter from where this data come from, administrative solutions, real-world data. Anyhow, all these issues are to be mixed, and clinical trials already use real-world data widely. The standards simplify working with the signals if there are enough instruments. And we need to ensure the correct processing of these messages. The first company that suggested the instruments for working with signals is Flex company and its software solutions. They help a regulator and the entire industry. And Flex solutions are used by pharmaca survey specialists. So we have heard already some speakers who are working with Flex instruments. Can it happen that Flex company will close its regional department? Then it is very dangerous and it shouldn't be done. The work with signals is conducted on all stages of the life cycle of the drug, but mainly this work is done on the post-registration stage. Of course, pharmacovigilance was born from real-world data. They always use real-world data in their work. Of great importance is the early period after registration of the drug for the first 10 years. This is the field for real-world data and real-world evidence, because the assessment of benefit risk is limited by a small number of patients and by a certain category of patients. By the time of registration, the drug shows only 50% of adverse events, and the rest of the 50% will be found later. Usually, the first, second, and third stage of the trial shows only the most visible adverse events. Mainly, these are allergic diseases. But in order to get the full information about all risks, we need to control these risks on all the stages. And here we can find rare and very rare adverse events, single out certain groups of patients. We can see the adverse interaction of different drugs. It is very important that all the interested persons would take part in collecting the information about the safety of drugs in order to cover this Achilles' heel of the pharmacovigilance. What are modern methods of working with the signal? So it starts with the message about the notice when the individual message is sent. So this notice about the suspicion for a possible signal happens at the very first stage. And of course, it happens on one of the three scenarios. First, it is getting the symptom of the adverse event into one of the three lists. It's important medical events, specific EMA list. The second scenario is serious adverse events. And the third is repeated adverse event for one and the same drug. The case card has the suspicion of the signal, and on the basis of the series of cases, we may make a conclusion about the appearance of a signal. So here we have quality methods of working with signals. They are based on the clinical assessment by a pharmacosurvey specialist. He or she considers the data for one or several cases. The user may form a series of cases to use them as an evidence base. This is a very time and effort consuming procedure that requires very good professional skills. And of course, it is done not only by doctors, physicians, but also people who compile files. This slide shows quantity methods of working with the signals that allow to make a statistic check of adverse events. These statistical methods are based on the different scales and networks. What methods are used? First of all, traditional statistical methods, methods of detecting the possible connection. Here we speak about proportional reporting ratio, about reporting odds ratio, and relative reporting ratio. And Roman figure 2 shows data mining methods, bias approaches. And here we speak about multi-item gamma Poisson shrinker and Bayesian confidence propagation neural network. If we speak about AI, we have a big problem here. The platform of artificial intelligence are not purely Russian, but they are imported goods developed by Russians. And when you want to find some national platform, it is not that easy. This slide shows approaches to the validation of signals, to the development of solutions. Using qualitative and quantitative methods, we validate information and make a decision about a signal. Are we supposed to study this signal further or not? And it is to be done by the doctor who knows this nosology very well, who knows comorbidities and all other things. If the signal is refuted, then we continue monitoring by standard methods of pharmacovigilance. If not, then we prioritize signals and create a signal card where the signal is described and assessed. This slide shows future promising methods of working with signals. The pharmacovigilance process is, of course, being perfected all the time. We have more and more exact methods of signal detection. And here I show you this new methods: adaptation of multi-item gamma Poisson shrinker, self-control case series, and information component temporal pattern discovery. Dear organizers, dear participants, in conclusion, I would like to say that working with signals is a very important process in the activities of every medical organization. It is quite natural that real-world data are used for making evidence based on this data.